Like speakers and so. Yeah, first.
All right, let's get this talk started. It is my great pleasure to introduce Dr. Sutherland today. I'm Ksenia, and I've had the pleasure of being Chandler's thesis advisor for the past few years. Chandler joined the lab as plant molecular biologist, and over the years, she's emerging as a computational biologist. I hope some of you can appreciate that. And she had the motivation and drive to do all of the research that she's going to tell you today. Um, she joined during the pandemic. We've seen each other on Zoom for most of her initial rotation, and we didn't know each other before from before grad school. And it was amazing. Like I was very pleased when Chandler said that she picked our lab against all over other labs, and it was absolutely amazing to see how she handled the research analysis, wrote her papers, went to conferences, presented her work at the conferences, responded to the community, and also towards the end of the PhD, actively reached out and brought new protocols, new approaches and tools to my lab, independently established new lines of research. And she's on the upward trajectory to continue that in Seattle, and that lab is also very lucky. Chandler is also a beloved member of the lab. Her energy and cheerfulness and can-do spirit is contagious. I think you can see it in the audience. When I told my son that Chandler is giving a finishing talk and he's invited, he's like, "Yeah, I'll be there." That's the evidence, and of course, that's the evidence to how influential and Chandler is on everyone. We did put a few tokens for you prepared. One is we made kind of like collage of some of the photos with the lab members and little of a card. So you can keep it on your desk and not forget us in the new lab.
Oh, that's really
And because we know, you probably have all the, everything, all the merch from Berkeley already. Kind of got your botanical gardens.
really sweet. Thank you.
Um, Chandler and I also we went to England and we visited Kew Gardens and we visited botanical collections there. And one of the memorable moments was when we saw cyanotype impressions of the botanical collections there. And so, Chandler, this is your own kit.
Nice.
You can impress a few Arabidopsis plants if you still have them or before you leave. And there are a few other things.
Thank
there.
you so much.
And so I'll give a floor to Chandler.
Okay. Thank you, Ksenia, for that really kind introduction. And thank you to everyone, family and friends, coworkers, for being here today. This was one of the most challenging talks to prepare out of my entire career because we have such a diverse audience, but I hope that there'll be something for everyone. And so for my friends who have not taken a science class or a biology class since high school, I hope there'll be something for you. And for the professors in the room, I hope there'll be something for you too. So bear with me as I guide you on this journey. So I'm looking forward to telling you about my thesis work, which, as stated here, is focused on the rate, context, and consequences of mutation in the evolution of plant immune systems. I know some of you may be thinking, plants have immune systems? But I think, here, let's see. You are all familiar with plant disease, especially if you've ever forayed into gardening. And so plants get diseases just like we do, and a lot of the same things that attack us attack plants. So plants can get bacterial infections, which is shown on the left here, fungal infections, viral infections, and they have a whole class of pathogens that we don't have to deal with, herbivores. So plants also recognize herbivory using some of the same pathways that they use to initiate their immune response. And that's a whole category of disease pressure that we don't have to worry about. But in the wild, plants are very healthy. They coexist and often in very dense environments, but for the most part, you don't see these types of immune or disease responses happening in a beautiful, happy, healthy forest or in very dense plant environments. So plants face diseases, but they're often very good at avoiding them, meaning they must have a really functional and successful immune system. And I like to think about immune systems in the context of the tree of life. So all organisms have immune systems, from bacteria to plants to fungi to invertebrates to ourselves. And all of these immune systems act in very different ways. They have very different mechanisms, but they share some of the same foundational goals. They need to recognize that they're being attacked, they need to mount an immune response, and then they need to stop that immune response once the attack is over. And so very different approaches, but a lot of the same goals for mounting the immunity and then returning it back to baseline. And one other key shared component of immune systems is the need for diversity. And all immune systems require diversity. And I like to think about this in this very classic hypothesis called the Red Queen hypothesis, which is taken from Lewis Carroll's Through the Looking Glass. It takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run at least twice as fast as that. So this was first applied to evolution with this idea that plants are constantly evolving and adapting to just continue to survive. There's no breaks in the game of evolution. And in the context of immune systems, this is particularly clear because you have to continue to evolve to recognize these diseases that are trying to avoid your recognition. And so when we look across the tree of life with this shared need for diversity, there's been a lot of really different and distinct strategies for how different organisms are able to achieve this diversity. So we're currently in the Innovative Genomics Institute, which is named such because of a lot of the work here that's done using CRISPR, which you may know as a gene cut and copy paste mechanism. It's a gene editing tool, but it was actually discovered through bacterial immune systems, where it's used to incorporate pieces of extra genomic DNA from viruses into the bacterial immune system to maintain an immune memory to future attacks. Ourselves as vertebrates, we have adaptive immunity. This is how things like vaccines work, where you're able to mount a new immune response to an unknown thing, where you can take a new pathogen and develop immunity to it on the scale of a few weeks. And this is pretty much specific to mammals and to vertebrates, but there's a lot of the rest of the tree of life that's not covered by these two mechanisms, namely plants, my favorite system of study. But there must be something, like there's the same required need for diversity, but we're not sure the mechanism of how plants are able to generate diversity at their immune receptors to keep pace with rapidly evolving pathogens. And the parts of the plant immune system I'll be talking about today are the receptors. So I'm interested in the first part of recognition of disease and recognition of pathogens. And specifically, I'll be focusing on NLRs. So I tried to go easy on you guys with the acronyms, but this is the one you have to remember. NLR means nucleotide binding leucine-rich repeat receptor, and they are the intracellular immune receptors of the plant immune system. And they have this tripartite architecture. So they have this modular architecture where they have a domain that's involved in sensing the disease, doing the actual recognition. They have an activation domain that keeps it on or off. And then they have an execution domain. So when it is turned on, it can go and enable the rest of the plant immune response, which includes a lot of transcriptional changes. There's plant hormones moving through the plant. The cell walls get thicker, the stomata close, antibacterials or antifungals are produced. It can be this like really layered and long-term response from this execution domain through a bunch of downstream pathways. And these NLRs, nucleotide binding leucine-rich repeat receptors, both directly and indirectly recognize pathogen-secreted effectors. So here we have a bacteria, it's landed on our plant cell, and it successfully evaded any extracellular receptors to then excrete these pathogen-supporting proteins into the cell. And NLRs either indirectly bind these or recognize that a disease is happening by guarding a plant protein and binding to that, or directly binding these pathogen-secreted effector proteins. So there's these kind of two mechanisms by which NLRs can recognize that disease is encroaching on the plant. And so zooming out a little bit, the main question of my PhD has been, how do plants generate new recognition specificity at their immune receptors to keep pace with these rapidly evolving pathogens and stay healthy, especially in these like group contexts? And I'll present today two stories, one looking at population level diversity and what that looks like when we map back rapidly evolving receptors onto an individual genome, and the other looking at mutations, which has become my obsession of the last few years, and looking to see how mutation accumulation and mutation bias informs the evolution of these receptors and the genome in general. And so starting with this first question. So when we think about plant diversity in these receptors, it's been appreciated for a very long time that NLRs are among the most diverse proteins across the genome. They have a lot of diversity at their receptor sequences. They seem to evolve very quickly. And we think that part of what keeps plants so successful is a population level diversity approach. So while we have a lot of diversity in our immune receptors within our bodies, plants are using more of a team effort where they have a lot of different receptors across a field of wild plants. And the diversity at that kind of population level is what's keeping plants so successful in defending against rapidly evolving pathogens. And NLRs diversify in lots of different ways, in the same way all genes do, but because they're so good at it, we like to zoom in on their mechanisms specifically. So single locus genes, they can duplicate, they can be deleted, inverted, there's point mutations, revivals, recombinations, domain fusions, and all of this is compounded by the fact that NLRs are very often organized into clusters. And that just allows for all of these diversity mechanisms to compound and become exponentially more effective. And so NLRs are evolving really rapidly from lots of different mechanisms, but today I'll be focusing on point mutation. So single changes in nucleotides and in DNA to create single base pair changes. And even within this one mechanism, we see a lot of diversity. And so this is Arabidopsis. I know Arabidopsis is not everyone's favorite plant, but it is one of my favorite plants because it's very easy to grow in the lab. It produces a lot of seeds. It has a very small genome, all of which make it very easy to study, but it also is a wild plant. And most of the other like very well-developed plant models are crops. And for this question, I really wanted to see what the like wild distribution of NLR receptors looked like. So I took the available DNA sequences of lots of different Arabidopsis NLR receptors. Well, actually, a collaborator did. And here's an example of one gene, but looking at all of the different protein sequences for that gene across the species. And it's very zoomed out, but each column represents one protein position. And we needed a way to capture all of this information as a single metric. And so we used Shannon entropy, which is this unbiased metric of amino acid diversity. So it's a way to look at all of this diversity kind of collapsed down. And so here's what's actually happening in the protein. You'll remember the execution, activation, and sensing domains of the NLR. And so the execution and activation, those are pretty stable. We need those to continue to work. And when we look across this alignment, you can see the rows are pretty neat and our Shannon entropy scores are pretty low. But as we enter into the sensing domain, that's under a lot of pressure to diversify, under strong selection for diversity, we see a lot more Shannon entropy. And so that is just showing that at this sensing domain, there's a lot of diversity and it seems to be evolving quite rapidly. So said collaborator is Daniel. And what he did was very clever in that he took this entropy at the 10th highest position. And we use 10 because 10 amino acids is the minimum amount to create a binding surface when we're thinking about antibodies. And if we look at all of the Arabidopsis NLRs across all of our sequences of Arabidopsis, like across the species, we get this distribution, which is roughly a bimodal distribution. And then we did this very mathematical and technical process of bimodal decomposition, where we split it in half and we called everything to the right highly variable NLRs and everything to the left non-highly variable NLRs. So these pink highly variable NLRs are evolving quite rapidly. They have a lot of diversity, especially at this or the sensing domain, while the non-highly variable NLRs are evolving more slowly. And a nice sanity check of this was that the indirect binders I mentioned earlier that are guarding their plant proteins were all non-highly variable NLRs, which makes sense because they should be evolving relatively slowly and stably. The highly variable NLRs included all of our known direct binders. So these are under more intense pressure to diversify and create new binding specificities. And with our two groups, I entered the lab and asked the question, are there any distinct features that differentiate these highly variable NLRs from non-highly variable NLRs on the genome level? So we see this high diversity across populations, but if we zoomed into one plant, one genome, are there anything that we can differentiate these two groups on that level? And the first thing we found was that highly variable NLRs are more expressed than non-highly variable NLRs. So these rapidly evolving immune receptors are on more often than the slowly evolving immune receptors. And when we look across the genome, we see if I ranked every gene in the genome by its expression, we see this enrichment of the highly variable NLRs in the most expressed genes across the genome. And this is true across different tissues. It was really robust as far as signals go. And when we think about this from a diversity perspective, expression without repair can be mutagenic. And so that was kind of our first clue that there might be something happening on the genome level to produce and maintain this diversity. We then looked at methylation, which is a carbon modification of DNA, and found that the highly variable NLRs were less cytosine methylated than the non-highly variable NLRs. And again, this enrichment was significant across the genome, while the non-highly variable NLRs look like everything else. Now, this one was surprising because cytosine methylation is actually associated with higher mutation. And so to see the conserved genes with higher methylation was a little bit puzzling. But in Arabidopsis, the higher or some level of cytosine methylation is associated with really stable genes that are expressed all the time and are really routine. And so there might be some unknown housekeeping mechanism of this cytosine methylation that the non-highly variable NLRs are enjoying while the highly variable NLRs are completely exposed. We also found that highly variable NLRs are closer to transposable elements, which are genetic elements that jump and insert and excise themselves from the genome. They're incredibly mutagenic in these incisions and exersions, but they also change the local methylation and expression landscape. And these highly variable NLRs are very close to transposable elements. They're among the closest genes in the genome to these transposable elements. So we were considering this as like a marker of their high variability. Even if I had a non-highly variable NLR and a highly variable NLR as neighbors, they retain their distinct features. So the non-highly variable NLR is not expressed. It has CG methylation. The highly variable NLR has a TE within its 5' UTR, within the gene sequence, and is highly expressed. And so it didn't seem to be like a specific chromatin state that was happening. It was like distinct to the variability character of these genes. And I think this was the most exciting result from this paper, in my opinion. So brace yourselves. But first, I looked at the nucleotide diversity, either one nucleotide diversity that changed the protein sequence, which is this high N, or nucleotide diversity that didn't change the protein sequence, which is this high S. And the highly variable NLRs were higher in both categories. But that makes sense. We differentiated them based on amino acid diversity. We expect nucleotide diversity to follow or precede. What was really interesting, though, is the ratio of these two variables, pi n over pi s, had no significant difference between the two slopes. And so this can be explained by a few things, but one of them is a higher rate of mutation at the highly variable NLRs, increasing both the non-synonymous and the synonymous diversity without changing the relative proportion of the two. With a collaborator at UC Davis, I also looked at the mutational probability based on chromatin state and found that highly variable NLRs have, on average, a higher mutational probability. And this is what really got me excited. I was like, okay, maybe there's both more mutation and then also like selective pressure, keeping this diversity high at the highly variable NLRs relative to the non-highly variable NLRs. Like maybe there is this mutational bias contributing to their difference in diversity. And so that was where we ended the first part of my work, which was we found these two different groups of NLRs, these ones that are evolving quite rapidly and the ones that are evolving slowly. By dividing them into two groups, we could make a lot of comparisons. And we found that the highly variable NLRs are highly expressed, they have low gene body methylation, and they're very close to TEs, and they have this increased mutational likelihood. So I've been talking for a minute. I'd like to pause and ask if there are any questions at this time. Carlos?
You're searching for something that getting back to these, and I missed it, right? So the NLR is basically like an immune system.
Yes, they're receptors. So they're the first line of defense where they're binding to the pathogen proteins to start the immune response. Can you go? Yeah.
Someone from Zoom, Zhentang, is asking, what is the biological mechanism that causes TEs to remain close to highly variable NLRs?
That's a great question. I don't necessarily think that there is a mechanism keeping them close, but our working hypothesis is that transposable elements inserting near a highly variable NLR might, or an NLR might make it highly variable by changing the local transcription and methylation landscape. Change.
Can you explain the normalized range that you were using for your X-axis?
Yes, I ranked every gene in the genome based on those three different categories, so expression, methylation, or distance to TE, and then I just highlighted where the NLRs were, both highly variable and non-highly variable, and then I used like a permutation test to see if it was significantly enriched one way or the other. Cool. If there aren't any other questions, I will. Final conclusion here is that distinct genomic features differentiate these rapidly evolving immune receptors from slowly evolving immune receptors. And I did the same thing in maize, but I won't show you the same data twice. Just know that I appreciate maize as a snack, as a staple crop, as a really impressive genomic resource. And I really loved working with it, but just trust that it was a good time. Okay, so kind of what I was alluding to is that the mutational likelihood was this loose thread, and it got a lot of pushback. When I first submitted this paper, I got this comment. Selection can drive allelic diversity, but I don't accept that variation in mutation rates in different parts of the genome can drive allelic diversity. And this comment made me very sad because Ksenia bought me a pudding to make me feel better, but we overcame it. And I do think with like the time of a few years away from this, it was like a very reasonable point in that we're hitting this point with population genetics and evolutionary biology and genomics, where we know that mutation is not random across the genome, but is that non-randomness adaptive? And that's a really difficult question to answer. And I think it's very fun to hypothesize that it might be, especially in the context of immune systems, but we simply don't have the data to prove that either way. And there's this hypothesis that's emerged of like a loaded dice. So mutations are not perfectly random like a normal pair of dice is, but maybe sometimes they're a loaded dice where you get a few more mutations in particular genomic regions every 1,000 cell divisions. And over millions of cell divisions and millions of generations, that is reasonable enough to then create the bias that we're seeing at these immune receptors. And so the other reason why I think this reviewer was maybe wrong, though, is that organisms, we know that there are these strategies for generating diversity across organisms, and there's self-mutagenic strategies that have evolved, which is a really risky evolutionary strategy. But when we think about both CRISPR and adaptive immunity, you're turning the mutagenic mechanisms onto your own DNA sequences, which can result in cancer if misregulated, can result in like disease and dysfunction. But it's so advantageous to have high diversity at your immune receptors that different mechanisms have evolved in different branches of the trees of life. And so I don't think it's totally preposterous to propose it's happening in plants. But so far, I've just been looking at observed diversity. I've been looking at mutations that exist in wild plants that have survived across the world. And what I really want to be measuring is mutation. And mutation is this combinatorial process of DNA damage and replication errors. It's influenced by epigenetic context. It's repaired by a lot of different mechanisms. And all of these things together create the distribution of mutations. But then those mutations are filtered through development and selection to this observed diversity. So it's very difficult to postulate about what's happening on the mutation level by using observed diversity. And so I needed to measure mutations. And mutations are very rare on the scale of genomes, but they're very common on the scale of organisms. And so here's a picture from the Botanical Gardens at Berkeley of a naturally occurring somatic mutation that has knocked out the plant's ability to produce chlorophyll in half the leaf. So it must have been a really early meristematic mutation. But these types of mutations, every cell in our body has mutations, every cell in a plant has mutations, but usually it's only in one cell. And so we have to be really accurate in how we quantify these mutations to be able to measure them. And one more analogy on how this works. If the genome is a book, all of the DNA in your cell is this book. You have to copy this book every time your cell divides for the however many cell divisions it takes to create a human or a plant or a tree. And I like to think about this copying process as more of like monks in a basement, like copying the Bible over and over and over again. And they're very accurate, they're very focused, but it's not perfect. They're not factory machines. It is like a living biological process. And so this led me to the question of, is mutation bias driving this diversity generation? And how can we quantify that? And so the challenge of measuring new mutations. There's been some recent developments in mutation sequencing that have allowed us to take the rate of error of sequencing below the rate of mutation for the first time. And so the way that this works is that there's been a lot of effort in reducing the errors that are introduced during the library prep process. And so you can get rid of a lot of errors just by not sonicating your DNA, but shearing it with restriction enzymes. And then the real like bread and butter of this technique is the ligation of strand-specific barcodes. So DNA is stranded, it has 2 halves, and you can take advantage of this by tagging each half individually. And then when you eventually sequence your molecules, you can go back and find from which original strand the DNA originated. So I only call mutations if they're present in multiple strands or multiple reads coming from the same strand and in consensus between the 2 strands. So PCR errors, sequencing errors, DNA damage, we can filter all of that out and get down to a reported error rate of 10 to the minus 9. So that's 1 billion. And so I've applied this technique to Arabidopsis. Here I'm showing a wild type plant, so a healthy normal plant, and I'm showing leaf, root, seed, and pollen mutation rates, and they're very low. So if our monk was copying, it would be 1 mistake out of every 100 million letters. So they're very good at maintaining a very low mutation rate. And there's not a huge variability across tissues. Like the leaf mutation rate is pretty similar to root, which is similar to seed, which is similar to pollen. And this is a big difference between the plant data and the human data because humans have a lot of variation in their mutation rates across tissues. And just to brag, the state-of-the-art way to study mutations before this technique was mutation accumulation lines, and they show quite a range in their estimates. And using this technique, we're able to get pretty tight estimates of mutation across tissues. We can also zoom in and look at the mutational signature. And so this is taking these mutations and looking at their mutation type. So from which base pair to which base pair they are mutating, and then where these mutations are happening. So these are all of the trinucleotide contexts in which the mutations are occurring. And this is what we call a mutation signature. It is reported to kind of represent the mechanisms of mutation that are happening. And so by comparing these signatures, we can see what the different mechanisms of mutation are, or at least see the different types of mutations and how they're shifting between treatment conditions or genotypes. And so here's an example of a genotype. This is a knockout of a DNA repair pathway. So I've disrupted the DNA repair pathway in this plant. And it has a much higher mutation rate. This one's 19 times higher than the wild type mutation rate, which would be like a really tired monk. He's on his 5th Bible. He's making 19 times more mistakes. And the mutation signature for this genotype is quite distinct from the wild type. We see this like striking C to T pattern, and we see this C to A bump, which is associated with oxidative damage. And we can begin to understand how changing this particular DNA repair pathway is changing the actual mutations that are accumulating. I was really interested, though, in immunity. So I gathered up a bunch of autoimmune genotypes. And so plants can have autoimmunity too. And these are all active in different mechanisms or different pathways of autoimmunity. So their immune pathways are activated in very different contexts and different types. But can anyone give me a guess on what these plants have in common, just visually? They are all green. I guess compared to like a healthy plant, a wild type plant. That would be my bad because I work in I'll take one more guess. They're small. Thank you. So all of these plants are really small, especially like MSH2 doesn't have a growth phenotype. So that's like I measured them at the same time, but this one is like big, happy green leaves, tiny, especially that one, like itsy bitsy little leaves. And so there's this phenomena in the plant defense or plant immune activation called the growth defense trade-off. So when you're actively defending yourself, you can't spend those metabolites for growing. And so all of these plants are spending all their time with an active immune system. And so they're all very small. They're very stressed. I had a strong hypothesis that they would have really high mutation rates, but that actually was not the case. All of these autoimmune lines had comparable mutation rates to wild type. Some even show slightly lower mutation rates than wild type. With the exception of uni, which is named after sea urchins for its bushy phenotype. And it does seem to have a higher rate of insertions and deletions than wild type, but there wasn't this like dramatic, every time you activate your immune system, you have a really strong increase in the mutation rate. And then I went back to my highly variable and non-highly variable NLRs. And this is just looking at the proportion of genes mutated. In the wild type, no significant difference. Autoimmune genotypes, no significant difference. And MSH2, no significant difference. But I don't think that my hypothesis is disproven yet. This is very few mutations to play with. Even though I get a lot of mutations out of this data, comparing mutations on a gene by gene level, you need a ton of mutations. And so I think that there is still room for an experiment where you directly compare these two and see if they're mutating more or less. But for now, we can't say that these NLRs are mutating more or less than each other. I did see my genomic features increasing these genic mutation rates, however. So here I've split up my mutations per gene by expression. So if the gene is turned on or not, here's the not expressed. And even beginning with very lowly expressed genes, they have a higher mutation rate. And as we move up expression, it's not like a strict linear relationship, but all expressed genes have a higher mutation rate than non-expressed genes. And looking at methylation, we see a much more linear relationship, but as we increase the methylation, we get an increase in mutation across genes that have high methylation. And both of these things were kind of already known and not too surprising, but it was really nice to see it reflected in this data. Expression, if you don't have coupled repair, is mutagenic. The DNA is in a single-stranded state. It's exposed to the environment of the cell. And if you don't have coupled repair, that will increase the overall mutation rate. And then methylation marks can increase your likelihood of spontaneous deamination. And so the more methylation you have, the more mutations you have. And so all this goes to show that mutations accumulate unevenly across the genome depending on genomic features. We know that highly variable NLRs do have different features. And so I still am hypothesizing that they may have different mutation rates, but I'm unable to give you that conclusive punch ending. And so second question break. Does anyone have any questions on the mutation half? Rebecca?
I'm curious actually with the result that there's fewer mutations accumulating in genes that aren't expressed. Usually those ones like the just sort of things like pseudogenes, which are genes, so do you have any idea why, but you aren't like accumulating mutations in those ones where these ones that actually are generating transcripts that they pop up?
Totally. Because I'm measuring somatic mutations, I think that there's very little selection happening at this stage. And so while there could be damage happening at those like I'm trying to avoid selection completely in this analysis. And so I think if I were to look on like a polymorphism level, you'd be absolutely right. But since they're not being expressed, they're just being like tightly bound up in their chromatin, nice and protected. So I'm not seeing any mutations in them. Ethan?
I guess kind of a follow-up. See when you were talking, these that are like critical for that. Did you survive?
I haven't looked at that exactly yet, but I expect that there is transcription coupled repair happening to keep those like highly expressed housekeeping genes like still functional. And I do have a transcription coupled repair mutant that does show like higher mutation in these transcribed genes than the wild type. So I do think that's what's keeping the cells functional. But yeah, really the only level of selection that's left is like the cell living long enough for me to extract the DNA from it. Yes?
So this is going to be a very much a layperson's version of. So I'm thinking only of the wild version of the neurosis, and wondering how climate change affects how basically an external environmental change affects the pathogens and the mutations that are caused, that are the response to the pathogens. Like, how are things changing in real time?
Great question, and what I want to study in my postdoc. So you've jumped ahead. But so climate change, we think, will make pathogen pressure worse. And there's a couple reasons why, but generally increased temperature and increased humidity increases pathogen ability to like enter plants and colonize them. And without a really strong winter killing off your pathogen, it's more likely to survive the next generation. And so climate change will absolutely make pathogens on crops and on plants in general worse. In terms of mutation, that's a very active area of research. My hypothesis is that heat can increase mutation rates, especially extreme heat, because the machinery that's involved in repairing DNA, some of those proteins are not as heat stable if you increase by like large amounts. Increased UV, which is also a climate change variable, does increase mutation rates. And so I do think that and also plants are locally adapted to various climates. And so as climates change, their ability to maintain stable genomes might not be as suited to their local environment. So yes, I think it will increase mutation rates and also pathogen pressure. Great, okay. So back to my original question, how do plants generate new recognition specificity at their immune receptors to keep pace with pathogens and stay healthy? I don't know, and I don't know if I've solved it, but I have an idea that I have generated through this work, which is the idea of anticipatory immunity. So bacteria have CRISPRs and lots of other related systems to put phage DNA into their genomes. We have adaptive immunity that's responding on the scale of weeks to new pathogens. I think plants, and I'm even extending this to other innate immune systems with multicellularity, fungi and invertebrates, have anticipatory immunity, where there's a higher likelihood of mutation at these immune receptors independent of an actual pathogen pressure. So there's this slightly higher bias towards mutation at their receptors. And once you do have a pathogen, you're slightly better suited to potentially having recognition. They're generating diversity kind of throughout their lives and throughout multiple generations and populations in anticipation of pathogen attack. And so if you've tuned out for the last 50 minutes, the takeaway I want you to have are that plants have immune systems and they're very successful. And that is thanks in part to diverse immune receptors. These diverse immune receptors have distinct genomic features that differentiate them from the rest of the genome. And then mutational bias contributes to this diversity pretty potentially, but I am hypothesizing that it does in a way that is really important for the success. And then a common question at these finishing talks that I decided I would just preempt is just, who cares? Like, why bother? And so studying plant immune systems have the very classic application of crops. Like, we eat a lot of plants, and so it's very important to us that crops are able to defend themselves against pathogens and that we are able to help them in this pursuit. And so potato blight is the classic example. Crop pathogens can be really devastating for yield and for populations of people. But also, we have this shared immune system problem of a lot of things attacking us and the need to generate diversity. And so I think that studying these diversity generation mechanisms across organisms will give us a better understanding of how our own immune systems evolve, how we can help our immune systems to not attack when they're not supposed to, and just generally understand better how diversity influences immune evolution and genome evolution. And with that, I'd like to start with my regular acknowledgement slide, and then I'll get into what you all came for like big acknowledgements in just a second. Any final questions on the science part of the talk? Cool. Okay. Well,